Welcome to another episode from the Veterinary Research Channel. The administration of blood products is a critical, often life-saving procedure for horses, but it is not without its risks. Approximately 16% of blood transfusions in horses are associated with adverse reactions. These negative outcomes can be triggered or made more likely by biochemical, structural, and immunological factors that change over time as blood is stored. These changes are often referred to as storage lesions and occur when blood is kept under refrigerated conditions, typically between 1 and 6 degrees Celsius, in plastic collection and storage bags. Storage lesions are time-dependent modifications to the structure and metabolism of red blood cells, ultimately increasing their susceptibility to breakdown, known as hemolysis. The study of storage lesions has been extensive in human transfusion medicine. Over the past few decades, advancements in metabolomics, the comprehensive study of small molecules within a biological system, have provided significant insights into the molecular changes that human red blood cells undergo during refrigerated storage. The power of metabolomics lies in its ability to simultaneously examine a vast array of molecules, allowing for a more complete understanding of cellular metabolism. This understanding can then be used to refine strategies for selecting blood donors, develop better additive solutions for storage, evaluate the impact of leukoreduction, a process of removing white blood cells, or identify quality markers to enhance the safety and effectiveness of blood products. Leukoreduction is a technique used to reduce the levels of white blood cells and platelets in stored blood. This removal is important because these cells can release bioactive substances, such as reactive oxygen species, products of lipid damage, and signaling proteins called cytokines, which have been linked to undesirable transfusion reactions in humans, including inflammatory complications and acute lung injury. In equine transfusion medicine, much of the focus has historically been on blood typing to ensure compatibility between donor and recipient. While there has been interest in comparing the metabolic responses of red blood cells to storage across different species, including humans, the available research specifically on equine red blood cell storage has been limited. Only a few studies have employed advanced biochemical techniques to investigate this area. These studies have revealed changes in the metabolism of equine red blood cells during storage periods of up to 35 days, both in whole blood and in concentrated red blood cell preparations. A thorough understanding of how equine-packed red blood cells are affected by storage over time is crucial for developing optimized storage protocols for this species. Therefore, the aim of the research we are discussing today was to use lipidomics, the study of lipids, and metabolomics to analyze both fresh and stored leukoreduced packed red blood cells, as well as non-leukoreduced packed red blood cells from horses. The goal was to characterize the metabolic profiles and how they change in relation to the development, extent, and progression of storage lesions in equine blood. To conduct this study, packed red blood cell units were obtained from 6 healthy blood donor horses enrolled in 2 different blood banks. Whole blood was collected from each horse using transfusion bags that included a filter for leukoreduction. The collected blood was then processed to create both leukoreduced packed red blood cell units and non-leukoreduced packed red blood cell units, which were then stored at a controlled temperature of 4 degrees Celsius for up to 42 days. Over this storage period, sterile samples were taken weekly from each unit for analysis. The researchers observed that in both types of stored red blood cells and the surrounding fluid, called the supernatant, there was a progressive accumulation of lactate products, while the levels of high-energy phosphate compounds, specifically adenosine triphosphate and 2,3-diphosphoglycerate, declined. They also found that hypoxanthine, xanthine, and free fatty acids built up in the stored red blood cells and their supernatants. Importantly, these detrimental changes were more pronounced in the non-leukoreduced packed red blood cells. The key finding of this research is that leukoreduction has a beneficial effect on the energy and redox metabolism of equine packed red blood cells, slowing down and reducing the severity of metabolic storage lesions. The results of the study clearly demonstrated that leukoreduction has a more significant influence on the metabolic characteristics of stored equine packed red blood cells than the duration of storage itself. Statistical analyses revealed a strong distinction between leukoreduced and non-leukoreduced samples based on their overall metabolic profiles. The leukoreduced units were characterized by higher concentrations of acylcarnitines, which are involved in fatty acid metabolism, while the non-leukoreduced units showed elevated levels of free fatty acids and carboxylic acids, which are intermediates of the Krebs cycle, a central metabolic pathway. Similar trends were observed in the supernatants of the packed red blood cells. The supernatants from leukoreduced and non-leukoreduced units formed distinct groups, with non-leukoreduced supernatants being richer in carboxylic acids and long-chain polyunsaturated fatty acids. In contrast, Leukoreduced supernatants had higher concentrations of short and medium-chain saturated fatty acids, antioxidants like urate, and metabolic fuels such as glucose, particularly in the early stages of storage. Furthermore, markers of protein breakdown and the metabolism of certain amino acids were more elevated in the supernatants of non-leukoreduced packed red blood cells. Looking more closely at energy metabolism, the researchers found that non-leukoreduced packed red blood cells consumed more glucose and had higher levels of glycolytic intermediates, which are compounds formed during the breakdown of glucose. Despite this apparent increase in glucose breakdown, they showed lower concentrations of important energy-related molecules like phosphoglycerate and adenosine triphosphate. Interestingly, there were no significant differences in the decline of 2,3-diphosphoglycerate levels between the two groups. In the pentose phosphate pathway, another important metabolic route, the non-leukoreduced red blood cells exhibited higher concentrations of metabolites in the oxidative phase, which produces molecules important for reducing oxidative stress. Consistent with this, the non-leukoreduced cells had significantly lower levels of reduced glutathione, an important antioxidant, and comparable levels of oxidized glutathione, indicating a state of increased oxidative stress. In terms of amino acid and carboxylic acid metabolism, the study revealed higher levels of succinate, fumarate, and malate, all carboxylic acids, in the non-leukoreduced packed red blood cells, along with evidence of increased glutaminolysis, the breakdown of the amino acid glutamine. Focusing on purine metabolism, which is involved in energy transfer, the leukoreduced packed red blood cells had higher concentrations of adenosine triphosphate, while the non-leukoreduced cells showed greater levels of adenosine triphosphate breakdown products. Additionally, the non-leukoreduced cells had higher concentrations of free guanine and guanosine monophosphate, suggesting increased breakdown of related molecules. The researchers also observed that leukoreduction significantly prevented the accumulation of polyamines, such as spermidine and spermine, which are arginine metabolites with roles in the immune system. Other changes in arginine metabolism were mainly related to the duration of storage rather than leukoreduction, with the exception of ornithine, which significantly increased in the supernatants of non-leukoreduced packed red blood cells. Finally, the most notable effect of leukoreduction was observed in the levels of fatty acids. Both in the red blood cells themselves and in the supernatants, leukoreduction helped preserve pools of acylcarnitines. In contrast, non-leukoreduced packed red blood cells showed an accumulation of long-chain polyunsaturated fatty acids and their oxidation products. Notably, short and medium-chain saturated fatty acids were found to be higher in the leukoreduced packed red blood cells. The findings of this study strongly suggest that leukoreduction exerts a more powerful influence on the metabolic characteristics of stored equine packed red blood cells compared to the length of the storage period. The results indicate that leukoreduction has a beneficial impact on several key aspects of red blood cell metabolism during storage, including energy production, the accumulation of carboxylic acids, purine oxidation, polyamine synthesis, and the preservation of carnitine pools, while also limiting the buildup of long-chain polyunsaturated fatty acids and related lipid damage products. Previous research examining storage lesions in equine red blood cells primarily focused on the red blood cell compartment itself, with limited information available regarding the surrounding supernatant. However, analyzing the supernatant is crucial for understanding processes like hemolysis, proteolysis, and the formation of vesicles, which are all part of the red blood cell's response to storage-induced stress. This study addressed this gap by investigating the metabolic effects of leukoreduction on both stored red blood cells and their supernatants. While the benefits of leukoreduction are widely recognized and mandated in many countries for human-packed red blood cells, there has been a lack of direct evidence supporting these benefits in horses. The metabolic storage lesions observed in equine-packed red blood cells in this study are similar to those seen in human-packed red blood cells, although the effects on energy and antioxidant metabolism appear to be less severe in horses. The researchers documented that leukoreduction helps maintain adenosine triphosphate levels, which is important because adenosine triphosphate fuels crucial cellular processes in red blood cells, such as maintaining ion gradients, membrane protein function for flexibility, the activity of protein degradation machinery, the proper distribution of phospholipids in the cell membrane, and ultimately, the survival of red blood cells after transfusion. Conversely, the lower concentrations of phosphoglycerate and adenosine triphosphate in non-leukoreduced packed red blood cells suggest an imbalance in energy metabolism, even with increased glucose breakdown. The higher levels of specific metabolites in the pentose phosphate pathway in non-leukoreduced cells suggest that increased oxidative stress might be diverting glucose metabolism towards the production of antioxidants, at the cost of overall adenosine triphosphate production. This idea is supported by the finding of higher ratios of oxidized to reduced glutathione in non-leukoreduced cells, indicating greater oxidative stress. The accumulation of most glucose breakdown products, but not adenosine triphosphate and phosphoglycerate, in non-leukoreduced cells points towards a potential blockage in certain enzymes involved in glycolysis due to oxidative stress. These observations are consistent with increased oxidative stress in non-leukoreduced units, as indicated by faster depletion of the reduced glutathione pool, activation of the pentose phosphate pathway, and increased breakdown of adenosine triphosphate. Mature red blood cells lack nuclei and mitochondria, the powerhouses of the cell, but white blood cells and platelets, which are removed by leukoreduction, rely on mitochondrial metabolism. The removal of these cells through leukoreduction likely prevents the upregulation of glycolysis that can occur as a result of mitochondrial dysfunction. This dysfunction is evident in the accumulation of carboxylic acids like succinate, fumarate, and malate in non-leukoreduced units. Notably, succinate has been associated with poorer outcomes in human transfusion recipients who receive large volumes of blood, potentially by activating certain receptors or indirectly promoting inflammation. The elevated levels of these carboxylic acids in non-leukoreduced packed red blood cells were accompanied by increased breakdown of glutamine. The study also revealed a strong protective effect of leukoreduction on purine oxidation. The higher adenosine triphosphate levels in leukoreduced red blood cells and the increased breakdown products of adenosine triphosphate in non-leukoreduced cells highlight this difference. Similarly, the higher levels of free guanine and guanosine monophosphate in non-leukoreduced cells suggest increased breakdown of guanine-based molecules. Interestingly, the expected increase in intracellular hypoxanthine, a common finding in stored human red blood cells, was not observed, possibly due to the activity of a specific metabolic pathway. In contrast, the antioxidant urate was depleted more rapidly in the supernatants of non-leukoreduced units compared to leukoreduced units. Consistent with signs of oxidative stress and mitochondrial issues, the accumulation of polyamines in non-leukoreduced packed red blood cells is significant due to their recently recognized roles in modulating the immune system. The presence of arachidonic acid and its derivatives in the supernatants of non-leukoreduced units could also contribute to platelet activation and inflammation. The increased activation of immune cells upon exposure to polyamines, succinate, and products of lipid damage might help explain the higher incidence of adverse transfusion reactions in horses that are not due to blood type mismatches. Leukoreduction significantly reduced the accumulation of polyamines. Other changes in arginine metabolism were primarily linked to storage duration, except for ornithine, which increased in non-leukoreduced supernatants, possibly due to the activity of an enzyme released upon red blood cell breakdown. The most striking and consistent difference between leukoreduced and non-leukoreduced units involved acylcarnitine levels and free fatty acids. Acylcarnitines serve as a reservoir for the activation of a metabolic cycle involved in maintaining cell membrane integrity under stress. The elevated levels of long-chain polyunsaturated fatty acids in non-leukoreduced units suggest an activation of fatty acid modification pathways driven by oxidative stress during storage, a phenomenon that leukoreduction appears to mitigate. The higher levels of short and medium-chain saturated fatty acids in leukoreduced packed red blood cells might indicate a reduced degree of vesicle formation, where small parts of the cell membrane bud off during storage. The researchers acknowledged some limitations to their study, such as the fact that factors like breed, housing, diet, and athletic activity of the horses were not specifically controlled. Given that genetic and environmental factors are known to influence the quality of stored human red blood cells, similar effects could be present in horses. However, this detailed characterization of equine packed red blood cells is a crucial step towards developing clinically relevant interventions in veterinary medicine. In conclusion, this research provides strong evidence for the beneficial effects of leukoreduction on the energy and redox metabolism of stored equine blood cells. It highlights significant metabolic differences between leukoreduced and non-leukoreduced units in pathways critical for immune regulation and red blood cell function. This study supports the broader use of leukoreduction in equine transfusions to improve their efficacy and safety. The application of metabolomic techniques in this context has significant potential, as it can inform the development of updated guidelines for equine blood storage and allow for further investigation into the impact of transfusion on clinical outcomes in horses. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.